In this video, you will see how to wash your Flowcell after sequencing using an Oxford Nanopore MinION device. For this procedure, you will use the following reagents: wash mix, wash diluent, and storage buffer. All these reagents come in the Flowcell wash kit from Oxford Nanopore. During the first step, you will use only the wash mix, WMX, and wash diluent, DIL, reagents. Place the tube of wash mix on ice for thawing. Do not vortex the wash mix tube at any time. Thaw 1 tube of wash diluent at room temperature. Mix the content of wash diluent thoroughly by vortexing. Spin down briefly and place on ice. In a clean 1.5 ml Eppendorf DNA low bind tube, combine 398 microliters of wash diluent and 2 microliters of wash mix. Mix well by pipetting. Do not vortex the tube. Keep the tube on ice. Before adding the wash solution, you will take the waste off from the Flowcell. Using a P1000 pipette, remove all fluid from the waste channel through waste port 1. Attention! During this step, it is vital that the Flowcell priming port and spot on sample port are closed to prevent air from being drawn across the sensor array area. Rotate the Flowcell priming port cover clockwise, so that the priming port is visible. Draw back a small volume to remove any bubbles from the priming port. You can do this by inserting the P1000 pipette tip into the priming port. To do so, set the P1000 pipette to 100 microliters, and turn the wheel until the dial shows 120 to 130 microliters, or until you can see a small volume of buffer entering the pipette tip. Load 400 microliters of the prepared Flowcell wash mix into the Flowcell, via the priming port, avoiding the introduction of air. To do so, release a drop above the priming port and, then, insert the tip onto the priming port, loading the remaining Flowcell wash mix. Please, keep a small volume on the tip, so you avoid introducing any air bubbles. Close the priming port and wait for 60 minutes. Ensure that the priming port cover, and spot on sample port cover, are closed. 5 minutes before the 60 minutes incubation is finished, thaw 1 tube of storage buffer, S, at room temperature. Mix contents thoroughly by pipetting, and spin down briefly. Again, if you prefer, using a P1000 pipette tip, remove all fluid from the waste channel through the waste port 1. As both the priming port and spot on sample port are closed, no fluid should leave the sensor array area. Rotate the Flowcell priming port cover clockwise, so that the priming port is visible. Draw back a small volume to remove any bubbles, by inserting the P1000 pipette tip into the priming port. Set the pipette to 100 microliters. Turn the wheel until the dial shows 120 to 130 microliters, or until you can see a small volume of buffer entering the pipette tip. Slowly. Repeating, slowly, add 500 microliters of storage buffer through the priming port. Take the Flowcell off from the MinION device. The Flowcell can now be stored at 4 to 8 degrees Celsius. Insert the protective cover into the MinION. Keep your MinION device in a safe place.